Hello, everyone, and welcome to our third weekly Lunch and Learn speaker series, all part of Parkinson Awareness Month. This April, we want to continue to help educate and raise awareness of the fastest growing neurological disorder in the world, Parkinson's disease. And we want to celebrate our Parkinson community. We are unshakable. We are now about midway through April, but there is still so many great activities planned for all of you. Spring socials around Alberta have been fantastic, and we still have roughly about 11 more to go. Please check our website to see if your social is upcoming, and if you have not registered yet, please do so. We really want to see all of you at the socials. For those of you in Calgary, we have a fun evening event planned coming up at the Comedy Cafe. Thursday, April 25th is a night of laughter, and the Parkinson Association receives a portion of all proceeds. If you love baked goods or treats, the Bake Pasteuria in Sylvan Lake and the Sweetery in Camrose have you covered, and they are donating a portion of all their sales in Alberta, in April, back to us throughout the month of April. Yoga Santosha in Calgary has karma classes every Saturday and are also donating a portion of their proceeds back to us. And Famblusa, I always say this wrong, Fablumosity in Edmonton is selling beautiful tulip arrangements all through April. And again, a portion of the proceeds are coming to us here at Parkinson Association. All of the details for all of these events are on our website. Also, during Parkinson Awareness Month, your donations will be matched dollar for dollar, thanks to the generosity of the Gordon and Diane Buchanan Family Foundation. So we have double the impact when you make a donation. If you have any questions during this webinar, please submit them in the chat below. At the bottom of your screen, there is a chat blurb icon. Click it, and then the chat will appear on the right-hand side. After the presentation today, we will try to answer as many questions as we can. Please note, this webinar is being recorded. All information provided in the videos by Parkinson Association and our featured speaker is furnished strictly for educational and entertainment purposes. This service is not intended to be diagnostic, prescriptive, or replace the relationship advice and/or the care of your physician. General questions about symptoms, treatments, available medications, complementary and alternative health care therapies, and current research will be fielded. Our guest presenter today is Terra Fairbanks. Her presentation today is Understanding Low-Intensity Focused Ultrasound for Potential Future Use in the Treatment of Parkinson's Disease. Terra completed her Bachelor of Science in Biology, focusing on neuroscience from the University of Victoria in 2022. During her undergraduate degree, she performed research on concussions and the spinal cord, fueling her interest to pursue graduate school. Following graduation, she joined the Gurgis Lab at the University of Calgary to complete her Master's of Neuroscience. Throughout her master's, she plans to investigate how a novel technology, low-intensity focused ultrasound, LIFU for short, may be used to alter the activity of deep brain regions. Her research will continue to contribute to the understanding of the LIFU in the hopes that one day it may be used as a therapy option for movement disorders such as Parkinson's. Thank you very much, and I'd like to welcome Terra.
Right, I'll just share my screen here quickly. Okay, hello everyone. Thank you for attending today's Lunch and Learn series. As I was introduced, my name is Terra, and today I'll be discussing the work that I've been doing with an emerging technique called Low Intensity Focused Ultrasound, or LIFU, to help develop it and understand it for potential future use in the treatment of Parkinson's disease. Now, before we get into the details of my study, I just want to start by going over some background information to ensure everyone is on the same page so we can all understand what's actually meant by low intensity focused ultrasound. So first of all, sound transfers energy through mechanical waves. And these mechanical waves can be heard once they reach the receiver, meaning the person or animal's ear. And they're called mechanical waves because they involve the physical movement of particles within a medium to transfer energy. And by a medium, I mean that could be a gas, a liquid, or a solid. And particles can also move in different directions, therefore forming different types of wave. The two main categories of waves being transverse waves and longitudinal waves. A transverse wave involves the particles in that medium moving perpendicular to the direction of travel. So say, for example, this arrow here is moving, or this wave here is moving to the right. That means that this red particle shown here would be moving up and down. And this creates a wave shown in this graphic. And a common example of a transverse wave in everyday life is ripples in water. So say, for example, you drop a coin into a still vessel of water, you'll observe ripples moving away from that point. And those ripples you observe are moving due to transverse waves. Longitudinal waves are the second type of mechanical wave. And these involve the particles within the medium moving parallel to the direction the wave is traveling. So say again, this wave is traveling to the right. That means that this red particle shown here would be moving left and right instead of up and down. And this produces a wave such as the one shown here on the bottom. And these are the waves that actually make up sound waves and therefore ultrasound. And ultrasound just refers to sound that is delivered at a frequency that's above the range of human hearing. And many of you have probably heard or had a personal experience with ultrasound before, as it is used for imaging, most commonly for imaging a baby during pregnancy. And this type of ultrasound is known as diagnostic ultrasound. In diagnostic ultrasound, sound waves are emitted from a device called a transducer, and they're kind of dispersed throughout the tissue, where some are reflected and captured to form an image on the computer, such as the one you see in this figure. But there's also another type and application of ultrasound called focused ultrasound, which I'll discuss today. Now, focused ultrasound is pretty much as the words described. It uses the same method as diagnostic ultrasound, where sound waves are emitted from a device called a transducer, such as the one shown here. But instead of the sound waves being dispersed throughout the tissue, they're instead focused on a very small area. And that area is called the focal spot. And within this focal spot, there's a very high concentration of energy. And if you were to actually apply that high concentration of energy to the brain or to neural tissue, brain activity within this focal spot will change, and it either can become more excitable or more likely to be active, or less excitable or less likely to be active. And this ability to cause both excitation and inhibition, depending on the settings of the ultrasound that you use, opens up the potential for ultrasound to differentially change activity of a variety of brain regions, and therefore has very high therapeutic potential to be used to treat a variety of different illnesses. But then we can take it one step further and talk about two different types of focused ultrasound. And these different types of ultrasound involve delivering the sound waves at different intensities. And those different intensities produce different effects on tissues and therefore have different uses. And by intensity, I'm just referring to different amounts of energy that are going to be provided within this focal spot. So if ultrasound is delivered at high intensities, meaning high amounts of energy are going to be delivered into the tissue, it's referred to as high intensity focused ultrasound, or HIFU. In this technique, application of high intensity ultrasound actually results in tissue death in that region. So essentially a little hole is going to be burned inside the brain at that very, very specific point. But it also can be administered at low intensities, in which case it's referred to as low intensity focused ultrasound, or LIFU. And in this technique, brain activity is only temporarily changed and no tissue modification or cell death results. And this is the technique that I will be discussing further today. But before we kind of go forward with that, I do want to discuss some other techniques that can be applied to change or cause activity of brain tissues. However, they all seem to have some sort of drawback to their use. And I'm just going to go over these techniques to highlight why it's important to develop low intensity focused ultrasound for use in clinical settings, and also why it has such high therapeutic potential. The first technique that I want to briefly discuss is deep brain stimulation, or DBS, which some of you may have heard of before, as this is a common surgical treatment option for Parkinson's. And in this technique, electrodes are surgically implanted into the brain into a very specific area of interest, which allows for very high precision of stimulation in the brain, and it also allows for deep brain structures to be targeted. However, because it does involve surgery, it is invasive and therefore increases the risk of infection and surgical complications. Now moving into the non-invasive techniques. Another commonly used neurostimulation technique is transcranial magnetic stimulation, or TMS. And in this technique, a coil is placed on top of the head over the area of interest, as shown in this figure, and magnetic pulses are delivered into the brain to stimulate or inhibit activity. And this is desirable. As I mentioned, it is non-invasive, so it does not require surgery. However, it does exert its effects over a reasonably larger area compared to that of deep brain stimulation, therefore has lower precision, and it also is not great at reaching those deep brain structures. A third neurostimulation technique that's commonly used is transcranial direct current stimulation, or TDCS, and this technique uses electrical currents that are sent by electrodes placed on the scalp to change the activity pattern within the brain. And TDCS has similar pros and cons to TMS that I just previously talked about, as it also is non-invasive, but it also still has reasonably low precision and can't target deep brain regions that well. So all three of these mentioned techniques, as you can see here, all have at least one drawback that limits their ability to be used as treatment options for a variety of different illnesses, which makes it desirable and almost necessary to find a technique that addresses these caveats so we can help advance treatment options and help as many patient populations as possible. And this is where low intensity focused ultrasound, or LIFU, comes in. Once again, this just describes ultrasound waves, which are sound waves that are delivered above the range of human hearing, being concentrated into a very small point and delivered at low intensities to result in temporary change of brain activity. And this technique actually addresses all of those concerns that I highlighted before with other neurostimulation techniques, and therefore has very high therapeutic potential. And it does this as it's non-invasive, eliminating the drawbacks of deep brain stimulation. However, can reach deep brain targets and maintain high precision, actually being able to target structures at millimeter precision, which other non-invasive techniques such as TMS and TDCS are not capable of achieving. And since LIFU is so appealing and does have such high therapeutic potential, many researchers around the world are trying to understand how it works and how it can change brain activity. But of course, with any neurostimulation technique, it also is important to note any side effects that might occur. I do want to note that side effects have not been seen to significantly differ from those of other non-invasive neurostimulation techniques, such as TMS and TDCS, which I just highlighted on the previous slide. But there still are chances that the subject might experience a headache, scalp discomfort, tingling, spasms, or twitching of facial muscles, neck pain, or lightheadedness. But all of these events should be temporary and they should resolve within a day. And there also are no previous reports of seizure with low intensity focused ultrasound. However, because it is a neurostimulation technique, there always remains a chance that a seizure might be triggered. Now, I don't want to get too bogged down in details, but I do think it's important that I just briefly introduce and highlight a small collection of work with low intensity focused ultrasound that have been completed by a variety of research groups around the world to truly show its potential and where research has come from to help me kind of paint the picture for how we came to study our research question. A lot of these previous studies have targeted structures in the outer region of the brain, known as the cortex, and application of this ultrasound to various regions have also caused a variety of changes in different study populations. So starting with an animal model, in rats, targeting an area of the cortex responsible for movement, which is shown here in orange, actually caused observed limb movements in response to ultrasound that were not present before ultrasound application. Also in humans, targeting that same region of the brain responsible for movement resulted in decreased reaction times. So that means that they had faster movement on a task following ultrasound than they did before ultrasound. And then just a third example, also in humans, targeting this area of the cortex that's responsible for receiving sensory information from our environment. So that would be like touch, sound, and sight, for example, integrating all that information actually resulted in improved sensory discrimination. And this means that they performed better when they were asked to distinguish between two sensory stimuli that were similar. For example, say you have 2 points touching your arm. And when they're farther apart, such as shown here, you can actually distinguish them as two separate points. But as they move closer together, they're going to eventually only feel like 1 point. And you can actually test this out on yourself as well. So in this study, before ultrasound, the 2 points, this, for example, was the distance of which the 2 points began to feel like 1. But after ultrasound, that distance decreased. And this indicates that the subject improved their ability to distinguish between two sensory stimuli that were similar. The studies targeting the cortex do show that ultrasound is capable of specifically changing brain activity. However, for it to likely have the largest effects in a Parkinson patient population, targeting areas that are directly involved in either disease symptoms and controlling disease symptoms, which mostly lie below the cortex, is likely more important. But targeting these areas below the cortex with focused ultrasound, or specifically low intensity focused ultrasound, have not yet been as widely explored, but these studies are becoming more common and a more popular area of research. Studies in humans targeting one of these deep brain regions actually has been observed with a decreased brain activity in that targeted area. Also targeting a deep brain region, again responsible for relaying sensory information from the environment, actually resulted in worsened performance on a similar sensory discrimination task that I described previously. So in this scenario, it would have been harder for the participant or the subject to distinguish between two sensory stimuli that were similar. So that would mean that they would have had a larger distance between those 2 points, where the 2 points on your arm would have felt like 1 than if it was before ultrasound. So overall, they had worsened performance when asked to distinguish between two sensory stimuli that were similar. And finally, just to throw in an animal study, a study that used monkeys saw that with low intensity focused ultrasound application to another deep brain structure actually resulted in altered decision making. And this was observed by increased decision accuracy with ultrasound application, but slower reaction times. And while these studies do show that low intensity focused ultrasound can be used to change the activity of these deep brain structures as well as superficial brain structures, there still is a significant amount of research that must be done before it can be used in clinical settings. And this includes understanding the impact that low intensity focused ultrasound has on deep brain regions, and if it's capable of causing changes in cognitive behavior and motor behavior in humans, as this still has not been widely explored. And by cognitive behavior, I'm referring to processes such as memory, decision-making, or attention. And then this is where my research comes in. So throughout my research during my master's, I am aiming to understand and investigate if low-intensity focused ultrasound is capable of causing a meaningful and measurable change in cognitive and motor behavior in humans. So specifically, I am looking at if low intensity focused ultrasound is capable of causing a change in performance on a decision-making task, where participants are required to inhibit a response when they're presented with conflicting information, as well as if ultrasound application can change movement speed when performing a motor task. And by doing this research, we're hoping to understand the parameters or the settings that are required for the ultrasound to cause a change in performance on these various tasks, as well as the possible effects that result from this novel technology. And we're starting experimentation in healthy subjects to ensure that there's no detriment in performance following the effects of ultrasound application before we move on to completion of ultrasound in a patient population. To do this, we're targeting a structure that's part of the basal ganglia, known as the subthalamic nucleus, or the STN, which is shown here in red in this image, as it is highly implicated in Parkinson disease. And it's also actually a very common surgical target for deep brain stimulation surgery. And the STN works to regulate motor function, and its activity is actually balanced against the activity of the internal globus pallidus, or GPI, which is shown here in green. And together, they work to allow for smooth execution of movements. And I know this is a lot of acronyms, so now I'm going to go back and break it down to their individual function and how they work together using some analogies so we can actually understand exactly what I'm trying to predict and what I'm trying to manipulate. So the GPI essentially serves as a gas pedal, and it works to promote movement. The STN works against the GPI and serves as a brake to stop or prevent movement, and the activity between these two structures are balanced. Now, in Parkinson patients, the STN is overactive, and that disrupts the balance between the gas pedal and the brake. And when the STN is too active, there's too much brake applied and not enough gas, which ultimately shifts the activity towards the brake. So you have too much input telling movement to stop, and you don't have much input telling movement to go, which ultimately causes movement to just overall be disrupted. And this contributes to symptoms such as akinesia or freezing and rigidity. But in healthy subjects, that balance between the GPI, or the go, and the STN, or the stop, is maintained, and this allows for smooth movements. However, if we were to manipulate that balance between the gas pedal and the brake, or disrupt it, theoretically, we should see changes in movement or performance of movement on given tasks. And we can actually achieve this by using low-intensity focused ultrasound. So applying low-intensity focused ultrasound to inhibit the STN, or the brake, means we would have less activity from the brake to say stop, and more activity from the gas to say go, should theoretically promote movement, as it is the opposite relationship that's seen in Parkinson patients. And since movement's going to be promoted, it should therefore cause faster movements, since we have no or limited or reduced activity from the brake. We also would expect that this should translate to performance on a cognitive task, as the STN, or the brake, also has cognitive involvement. And this is the task that I'm using to look at decision-making, requiring movement to respond to the correct choice. And if the participant is moving faster, we would predict that their response time on a task also would become faster. And if they're responding faster and not taking as much time to consider the correct response, we also would predict that the error rate would increase. And this is what I'm aiming to achieve in my current study. To test this, I'm employing two different tasks. We're using a motor task, which is a finger tapping task, and we're using a cognitive task, which is called the Stroop task, which is a color naming task. In the finger tapping task, I ask the participants to place their hand flat on the table, as shown in this image, and tap their right index finger as large and as fast as they can for 20 seconds. And they're prompted to start and stop by tapping cues on a computer screen. We also attach a brace to their finger to prevent their finger from moving while they're tapping, just so it's consistent between participants, and also an accelerometer on the end of their finger to record their movement. And I've included an example recording here at the bottom. And then from this recording, I extract the frequency of finger tapping, or the number of taps per second, for analysis to see if there's been a change in motor performance following ultrasound application. The cognitive task that we selected is the street task. And in this task, participants are presented with color words, either the word red, green, blue, or yellow, and either the same ink color or an opposing ink color. So this could include the word red being presented in red ink color, or the word red being presented in blue ink color. And participants are presented with this word and are required to respond to the color of the word instead of the meaning of the word by tapping the corresponding key on the keyboard. So for example, say the word red was presented in red ink color, they would be required to press the red keyboard key. But if the word red was presented in blue ink color, they'd be required to tap the blue keyboard key. And in this task, I record how fast they respond to each color word, and I use it to look at how their response time changed with ultrasound. Then I also calculate something that's known as the Stroop effect, which just tells me about their ability to suppress a response when presented with conflicting information. And then I also look at the number of errors that they made to look at overall error rate, and I compare their performance on the control or no LIFU condition and the ultrasound application or LIFU condition. But before I can actually start recording data, I have to complete a series of planning steps to ensure that we're actually going to hit our target with ultrasound, and that the ultrasound settings that we're going to use are within the safety requirements as recommended by the FDA and Health Canada. So participants first come in for an MRI scan. We perform 2 structural scans, which are shown here in the left and in the middle. And we use these just to visualize our target, so we can actually make sure that I'm placing the point I want the ultrasound to hit correctly. And then we also perform a scan shown here on the right, which images the skull bone. And this is important as we need to understand the composition of the skull bone, so I can accurately predict and simulate how the ultrasound is actually going to pass through the skull and impact brain tissue. I then start by locating my target on the MRI. Again, this is at the STN, which you can actually see in this little red box, and you can actually see the nuclei here as well. And then I adjust the trajectory that I wish the ultrasound to enter the brain at. The approximate trajectory is shown here by this black line, and we selected this trajectory and these approximate angles as we hope to hit the STN along its longest axis. So as you can see here, this is just a graphic of the subthalamic nucleus and its structure in the brain. It's kind of a lens shape, so we hope just to skewer the longest axis of the STN there, which these approximate angles can achieve. I then run the simulations, and these serve to model and provide a prediction of the possible mechanical or the physical effects on brain tissue, and also any potential temperature rise within the brain, so we can assess the safety of applying ultrasound at different intensities. As if we have too many mechanical effects, or if the temperature rise is too high in tissue, it can actually cause permanent tissue damage. And then once the planning is complete, we then can move on to our experimental sessions. I'm performing the experiment double blind, and this means that the participant and the experimenter, myself, are not told which treatment they're going to receive on a given day. And participants come into the lab on 2 separate visits where we randomize and either administer a control, so no ultrasound, or the real ultrasound treatment. And then we start by performing a practice of both of the 2 tasks that I just described. And this just serves to ensure that the participant understands the task and the order that they have to be performed in before they actually proceed to take baseline measures. And participants perform these while they're comfortably seated in a chair. And the baseline measures and experimental measures involve alternating between the finger tapping task and the color naming task. So each is completed 4 times with a 1-minute break in between. And then I set up the device that delivers the ultrasound. And I start by attaching a tracker by either glasses or headband to the subject's head. And then these fiducials, these little balls on the end of the tracker that you can see in this image, allow me to know where the participant's head is and the orientation of their head in space in real time, so I can accurately move the transducer or the ultrasound device to hit our target. And the transducer is this big black thing that you can see here in the top of this image. To tell the tracker where the participant's head is, I have to first register where that tracker is in relation to specific landmarks that I placed on the head. To do this, I place a pointer on the nose, between the eyes on the nasian, on the left ear, and on the right ear. And then I validate by placing the pointer on a variety of locations on the top of their head. So I can make sure that the camera or software, where the camera or software thinks the head is actually where the head is. So we can ensure that it's highly accurate when I'm positioning the ultrasound device to hit our target. And then we secure the participant's head with a chin strap holder and a little stabilizer on the back of the head, which you can't see super well, but you can see the chin strap here, to make sure that their head doesn't move and moves very, very, very little throughout the experiment. And then I move the transducer or the ultrasound device into place using this mechanical arm, and the final position resembles the one seen here. Again, this is the actual ultrasound device that we're using. And then all of these gaps between the head and the transducer, we fill with a lot of ultrasound gel. And that just served to make sure there is a consistent medium for those sound waves to travel through for us to accurately hit our target. And so that all the energy is actually delivered to our target. I then run the sonication or the ultrasound. And this here shows 3 different orientations of that focal spot. So that concentrated energy from the ultrasound in a simulation for this particular participant. And the ultrasound will mimic this. So the red shown here is the area of the brain that will see the highest intensity of ultrasound or the most energy. And then around the outside in the blue, that's the area of the brain that will experience the weakest energy. And anything that isn't part of this will not be experiencing the effect of ultrasound. Then once I've delivered the ultrasound, we then rerun the experimental measures, which follow the same procedure as baseline. And then I perform an analysis to compare how they performed after treatment to their baseline measures to account for any day-to-day changes that might have occurred, such as accounting for fatigue, or maybe they didn't eat as much that day, their just overall focus was off, in order for us to actually observe if their performance at any of the tasks changed in response to ultrasound compared to the control. Now, before I jump into some very brief results, just to go over the trends that we've observed, I just want to reiterate the tasks that we are using as well. So to test motor performance in relation or as a result of low intensity focused ultrasound application, we're using a finger tapping task where the participant is required to tap the right index finger as large and as fast as they can for 20 seconds. And then I record their movement with an accelerometer and calculate their frequency of finger tapping or the number of taps they perform per second. And we see that in this motor task, the number of taps per second or the frequency performed between the control and the ultrasound condition did both decrease, so they overall tapped slower. However, the changes between the conditions and the changes from baseline are so small that there's likely no change in finger tapping frequency that resulted from ultrasound application in healthy subjects. For the cognitive task, again, this is a color naming task to observe if we can alter decision making and the ability to suppress unwanted responses to those conflicting information scenarios. Once again, in this task, the participant is presented with a color word in either the same or the opposite or a different ink color. And they're required to read the word but respond to the color of the ink rather than the meaning of the word. So for example, the word red in red ink color, they press the red keyboard key, whereas the word red in blue ink color, they press the blue keyboard key. And something that I didn't mention before, but I'll mention now, is just that the condition where the color of the ink and the meaning of the word are the same is the non-complicated condition, and the condition where the color of the word and the meaning of the word are different is the complicated condition. And then throughout this task, I record their response time, so how fast they responded, and the number of errors that they made, meaning either incorrect responses or missed responses. Starting with the errors made, we see on the non-complicated condition, that is where the color and the meaning of the word are the same, that both the control condition and the ultrasound condition resulted in a similar number of errors, which you can see by this little red slice here, and also the similar error rate. Also, overall, as you can see by this ratio of the blue being correct responses, red being incorrect responses, that participants just performed very well with both conditions. And this just suggests that there's likely no meaningful difference in errors observed between treatment conditions. Moving on to the complicated error rate. So again, this is where the color of the word and the meaning of the word are different. We'd still see similar results. Overall, participants just performed very well. We have similar errors made in the ultrasound and the control condition, also similar error rates. And this suggests that there's just no meaningful difference in error rate on complicated trials that resulted from ultrasound application. We do, however, start to see differences when we look at response times. For the non-complicated trial, where the color of the ink and the meaning of the word are the same, participants responded faster following ultrasound application than they did following the control condition of no ultrasound administered. And this suggests that low-intensity focused ultrasound application may be associated with faster response times on non-complicated trials. And these results are even more apparent on the complicated trials. Again, this is where the color of the ink and the meaning of the word are different. And we see that following ultrasound application, there seems to be an amplified effect than following control. And this again suggests that low intensity focused ultrasound might be associated with faster response times on complicated trials. Now, the Stroop effect is our result that really highlights the difference between the control condition, so no ultrasound administered, and the focused ultrasound condition. As a reminder, the Stroop effect allows us to assess the participant's ability to suppress a response. So to suppress those more natural responses when presented with conflicting information. And looking how the Stroop effect changed from baseline allows us to observe the relationship of complicated and non-complicated response times to really show us the effect of ultrasound administered to the STN on cognitive performance. So, following the control condition, which is shown here in blue, we see that the Stroop effect increased. But following the ultrasound condition, we see that the Stroop effect decreased. And this is interesting, as if you recall from the response time results, following both the control and the ultrasound, we see some degree of faster responding. But here it's indicated that the relationship between the complicated and the non-complicated stimuli is different between the control and the ultrasound conditions. And if we look further into why that is, we see that in the control condition, participants overall responded faster on that non-complicated condition from baseline than they did on the complicated condition. Whereas in ultrasound, that relationship is flipped. And we see that people responded faster on the complicated condition when compared to the non-complicated condition. And this is interesting as it does show that the participant overall spent less time responding on the complicated stimuli compared to the non-complicated stimuli with ultrasound, which was not seen in the control condition. And this is actually what we would expect from inhibition of the subthalamic nucleus or the brake, suggesting the activity of the brake was likely suppressed with low intensity focused ultrasound. If you remember, the GPI or the gas promotes movement, whereas the STN or the brake inhibits movement. So if we were to suppress activity of the subthalamic nucleus, we will get more activity from the gas, which in turn should lead to faster response times. Specifically, we would expect a faster response time on the complicated condition, as they're responding faster on the trial that would usually take more time to consider the correct response, which indicates they have altered decision-making, and as they're not taking the same amount of time to consider the conflicting information in the complicated condition. And these results have also been supported by other primary literature or other studies in Parkinson patients who have received deep brain stimulation to our same target, the brake. And when stimulation is on, they see that same trend of faster responding on non-complicated trials, faster responding on complicated trials, and a decreased Stroop effect. And these are the same results that I presented here in our research. However, it is important to note that Parkinson patients do have a hyperactive STN and therefore are not a direct comparison to our study population, but it still does give insight into what our results may say. Previous research also has shown that deep brain stimulation to the STN or the brake is also associated with an increased errors made on the Stroop task. But we have not observed this so far in our study. This is not consistent with results that we have seen. And this just tells us that likely the task that we're using is capable of capturing those changes in response time. However, it just is not sensitive enough to actually detect any changes in errors in healthy subjects. But this may change with the patient population. So first of all, just in summary, we saw no meaningful changes in finger tapping frequency between conditions. And this therefore suggests that low intensity focused ultrasound did not have any impact on motor performance in healthy subjects. For the cognitive results, we did not see any indication of a meaningful change in error rate, as overall participants just performed very well. But the response times revealed that following ultrasound, participants did appear to respond faster on both non-complicated and on complicated trials than following the control. And this was further exemplified in the decreased Stroop effect following ultrasound, but increased Stroop effect following the control condition, highlighting that overall participants spent less time on the complicated trials with ultrasound. And these preliminary results are exciting, or they're quite exciting, as in combination with other primary literature, they do suggest that we were able to change the activity of the subthalamic nucleus or the brake with low intensity focused ultrasound. And this change was translated to a measurable change in cognitive performance. So overall, this research does show that low intensity focused ultrasound can target and change the activity of subcortical structures, which can be measured by a change in their output. But still, many unknowns remain of how we can actually optimize the use of low intensity focused ultrasound in a clinical setting. And this includes, we still are completing this current study as we're right in the middle of data collection at the moment, as none of our results have actually been seen to be statistically significant, meaning we need to have, so we'll continue with more participants. However, the trends that we have observed here can still give insight in the ability of low intensity focused ultrasound changing the activity of those deep brain structures. Deep brain stimulation, which I briefly introduced at the beginning, is also a well-established treatment for Parkinson's disease. And the field has a very good understanding of how electrical stimulation affects brain structures and how it can be used for treatment of movement disorders. Therefore, if we apply ultrasound to a patient population and we compare its effect to that of deep brain stimulation, it can tell us or allow us to understand how electrical stimulation will equate to ultrasound stimulation and the possible parameters that we need to use in ultrasound to acquire similar effects as deep brain stimulation. And this is what the laboratory that I'm a member of is aiming to do after my current study is finished to advance the future of low intensity focused ultrasound. And with that, I hope that I have convinced you that low intensity focused ultrasound is a very promising future therapeutic option, as it is effective at targeting a variety of different brain regions. It's also non-invasive, and it also can target deep brain structures. But before it can actually be considered to be used in a clinical setting in patients with Parkinson's, further research is still required. And my research presented here does contribute to this foundation for further development and further studies in patient populations. And with that, I'd like to thank my supervisor, Dr. Gergis, and co-supervisor, Dr. Pike, as well as my committee members, the medical student who has been helping me out, and all of the members of the Neural FUS lab at the University of Calgary who have helped with my research so far, as well as all of my funding sources. Thank you to all of you for attending this Lunch and Learn seminar, and I'm now happy to take any questions that there might be.
And pardon me, thank you very much, Tara. That was super exciting and super interesting. And I will open up the chat if there are any questions. Okay, so here's a question. Is there a risk to destroy or kill any good brain cells during this process?
During the low intensity focused ultrasound, since there's no permanent changes to the cells, it's all temporary, there's no risk. We make sure, that's why we run all of the simulations and all of the planning before to ensure that we won't have any structural changes or any permanent changes result from the low intensity. The high intensity one I don't work with, so I'm not as familiar with it, but I do know that they do actually cause cell death. So in that case, I would assume that there is a potential, but again, I'm not too familiar with that technique as I don't work directly with it.
Is your study still recruiting
Yes, we are.
participants? Okay. And how would someone go about getting in touch with you, or what would be the process for them to become involved as a participant?
There's a variety. Probably the best way would be to send me an email. And then we have a series of inclusion and exclusion criteria that I have to run through with you to make sure you are qualified. But if you are interested, you're more than welcome to send me an email. I can share it at the end of this as well. And then we can be in touch to chat about participating.
There is another question that says, I understand DBS can only be done once. Can ultrasound be ongoing?
Yes, it can. So that's one of the part of the low intensity ultrasound that we're still trying to understand, is that we don't exactly know. So we know that the changes from low intensity focused ultrasound are temporary. One of the other studies in the lab that I collaborate with just did a study, and they saw that the effects last upwards of an hour. So our idea is that if you come in for several treatments in a row, you might have more prolonged effects. So, but they're still temporary, and low intensity is still being developed. So you can get it done more than once, and it doesn't cause any structural changes.
Are there any other questions? I don't see any in the chat. How much longer is this study going on for?
My current study, we're hoping to recruit about 10 more people. So we're hoping to be done kind of end of June. And then we'll be starting the second part, but we are still kind of working out the details of that. We haven't completely decided with that. but then that will be starting probably in the fall sometime.
Is this research only in Calgary?
Yes, it is.
Okay.
There's other institutions that are working elsewhere, but the study that I am working on is only in Calgary.
Okay. Is it an exclusion criteria if people don't live in the Calgary area?
It's not an explicit exclusion criteria. However, I'm not too sure how the lab would go about compensating travel charges.
Okay.
Because it does require a couple different visits into the lab. You have to come, first of all, your MRI, and then we have 2 visits that you have to come in as well, spaced about, the first one's about a month before, and then the other ones are spaced minimum 24 hours apart. So it does require a bit of a commitment. So I'm not too sure. That would have to be something that I discuss further with my supervisor.
Okay. I have a question. How advanced are your patients?
So in my study, we're doing this first of all in healthy participants, but then the next study is going to be in Parkinson patients who are eligible for deep brain stimulation surgery. So the idea is that they'll come in before the surgery and we'll do this ultrasound and we'll take all of our measures, and then they'll go and complete the deep brain stimulation surgery. And then we will do the measures again after and compare them. So I'm not a physician, so I'm not sure about like the qualifications for deep brain stimulation surgery. But they'd be decided from a panel of neurologists and neurosurgeons for their eligibility.
This question, I assume that this stimulated area in the brain will only be temporary. How can the good results be long lasting?
That's something we still are not super sure about. This is a very new technique. So our thought is that potentially, if you have sessions that are spaced apart, you could have more long-lasting effects. But that's just kind of a theory right now. We're not actually sure. So we still need a lot of research to figure out like the dosing of it and the different settings we should use and the effects and how it might affect different patient populations, and all of that still needs to be worked out, if that answers your question.
It's complicated.
Yeah.
Yes. But again, it just shows us how important research is, because these are emerging potential technologies that people could potentially be involved with.
Yes, exactly.
Yeah. Are there any other questions? I will. Well, if there are no further questions, oh, just a moment. Will you test people when they are on or off?
By on or off, are you referring to the stimulation or
I
like
think on or
medication?
off medication. So if
Oh,
they're, yeah,
um, that isn't,
if their meds are working or their meds are not
oh,
working.
meds are working or meds are not. That is also something that I am not sure about. We haven't discussed that quite yet. That would be something that in the next couple months, I'm sure we will discuss. And that would again be kind of a decision by the neurologists and neurosurgeons that are involved as well. I'm not sure the relationship of, to my understanding, and this might not be correct, is that deep brain stimulation often is useful for patients who have progressive Parkinson's that are not responding as well to medication. So in that situation, it's going to be more that population that we're targeting instead of the population that other treatment options are working for. As I know, deep brain stimulation is a pretty extensive procedure to get done. So not everyone who has Parkinson's can just get it right away. So, but that would be kind of at the discretion of the neurosurgeon and the neurologist that we're working with.
Right. And just like there is very strict criteria for deep brain stimulation, there will be strict criteria for ultrasound as well.
Yes.
Yeah.
And
So
I'm
it's
not too sure what those criteria are right as of right now.
and it may fall into that category of a potential advanced therapy where we're looking at whether or not there are other potential options to enhance the current therapy or to make it so that that therapy is lasting.
Exactly. Exactly. And there's also other studies that are going on. And ultrasound is also actually being used to help open up the blood-brain barrier. So within the brain, you have a collection of blood vessels surrounding it, which limits different things entering the brain. So whether that's like nutrients or in our case, we'd be interested in like drugs. So if there's certain pharmaceutical agents that you can take, ultrasound can actually be used to enhance their entry into the brain, which is another possible therapeutic application. We're not studying that in our lab, but I do know that that is under development elsewhere as well.
Yeah. Well, I think this was fantastic. And again, I just want to say thank you very much for sharing your research with us. And for those of us that you joined us online, thank you for joining today's webinar. I think it's been fantastic that we get to see young people like yourself that are excited in advancing research in potential Parkinson treatment. So thank you again, Tara, very much appreciated.